Are those the light up boys?
Yes, they're the ones with the little yeah, like light up probe. They fish for other fish. And so we're doing cell reproduction, so we kind of talk a little bit about reproduction in general. It turns out that those fish have a really interesting reproduction strategy where the organism carrying, like with gonads, or not with gonads, but with testes, actually bites another organism that has ovaries, and then they like become one organism and which has both testes and ovaries, and so they can self-fertilize whenever they feel like it. So didn't know that happened.
Isn't that why one of them is like significantly smaller? Like the big angry ones that we always see are the females, and then the males are just like basically little tiny like
Yes, I had no idea.
sperm sacs with fins basically.
Yes, I had no idea. Actually, if you look, yes, I'm so glad somebody else knows. If you look at a picture, I'm going to share my screen, of an anglerfish, the majority of the time you see both the male and the female, because the male is literally like an appendage on the female. And they actually cannot reach sexual maturity until this happens. So I was just honestly like, I thought that was one of the coolest things I'd ever heard. And I really, really loved prepping the lectures for next week because of it. So we're going to talk about anglerfish next week. They look terrifying, and they live really, really deep down in the ocean. And so I actually thought this was really interesting because it's sort of like, again, I like to ask why, like why? But deep down in the ocean, it's so rare that these organisms would encounter each other. So it makes sense that if you find a mate, you just, never leave, physically never leave each other. It is the best way to get, to enable reproductive success in that species, which is fascinating. Yes, it was in Finding Nemo. Yes, I can show you again. Let me show you a preview for next week. This is our slides for next week, and yes, I did include Finding Nemo because of it. Yes. And also this just crazy picture right here. So I just thought that was so fascinating. So I will fully admit to you, normally I am kind of like, I don't really want to talk about mitosis and meiosis. But then I am now very excited about it. So how does it fit in? I will tell you next week in the lectures. But I just made it fit in. So I talked about what I wanted to talk about, anglerfish. So yeah, super fascinating. So that's what I've been, oh yeah, we'll talk about clownfish too, because clownfish also exhibit what is called, it's not temporal, even though that's what I want to keep talking about it. They exhibit what's called sequential hermaphroditism. So clownfish switch from having ovaries to testes over the course of their lifetime. And it all has to do with just like the timing, their development. So yeah, every clownfish in its lifetime will have both testes and ovaries. So pretty fascinating stuff. Just like super cool. I love that stuff. Okay. So yeah, Finding Nemo is, I love, we'll talk about a lot of Finding Nemo characters, I guess. Oh, I just saw the rest of it. Nemo was supposed to raise an awareness. I know, that is true. Yeah. That's why I would say like the majority of cute animal movies. I mean, every movie we see, my daughter wants an animal from that movie, But luckily, we have been watching a lot of Inside Out lately, and I convinced her that I am Joy. So she got her pet out of that movie, and it's her mom. I am Mama Joy.
That is so cute.
It's really cute. It's cute. It worked out very well because we are not going to get a pet anytime soon. Okay, so anyway, I'm so glad you're all here. I hope you're all doing well. Yeah, it's a good, it's a good time to check in, know I'm here. But before I get started on anything, okay, so I will tell you, my other class told me that they did not feel comfortable with transcription and translation. So they chose to talk about transcription and translation instead of kind of clarifying anything to do with genetic regulation. I do want to assure you that if you have any questions about genetic regulation, two things about that. One, genetic regulation, as far as like how well you need to understand it, I would say that I would encourage you to focus on transcription and translation. So genetic regulation is important, but as for like the questions on the review and the checkpoint and what we need you to get out of this class in general, I would say that the processes of transcription and translation are, yes, I will talk about histones, are kind of like where you need to focus your attention. So because of that, I really want to make sure that I can hit what you need. I do want to mention that I did open the Unit 4 review. So it opened yesterday morning, and it won't close until next Wednesday. So on your recommended schedule, I recommended just taking a review attempt or your first review attempt. If you have time, maybe on Sunday or, maybe tomorrow, whatever works for you. But I would recommend doing it maybe before you submit your muddiest points, because then your muddiest points for this week, if you would like it to, can focus on like what you saw being heavily represented on the Unit 4 review. So if you saw like, okay, I guess I didn't need to know too much about this, I need to know more about this, it'll just help you kind of know where to focus your attention. And then we'll be meeting on Tuesday, so we can talk about that before you're even done with all your review attempts, and the checkpoint won't even open until Thursday. So that means we'll have a lot of time to talk about what is on the Unit 4 review before you actually take the Unit 4 checkpoint. I also want to assure you, so the stuff that was covered this week, which was like cell cycle and cancer, that is not on the Unit 4. So Unit 4, the review and the checkpoint, is only transcription, translation, and gene regulation. There may be some very light questions kind of discussing DNA replication, which was covered this week, but I want to say maybe there's like 2 questions, and it just has to do with the fact that both transcription and replication rely on complementary base pairing. But aside from that, the review, the checkpoint that is coming up is only transcription, translation, and regulation. So not this week, but the 2 weeks prior to that. So you can see that, again, you get 3 attempts on the Unit 3 review. So if you wanted to take one of those soon to see kind of what to expect, I would recommend it if you have the time. It's open notes, open book, open resource, so you can really kind of guide your studying that way. So I want to know, like right now, where you're at in terms of what you need help with. So Layla mentioned histones. We'll absolutely talk about histones. I did create a little poll just to kind of like gauge. So hopefully, can everybody see a poll? if you would mind just like answering that poll for me, that way I can kind of gauge what we want to focus on. And I'll give you like a minute, if you don't mind. Or until we reach 100%. Okay, so let's see. All right, if you haven't yet, please cast your vote. I'll give you a couple more minutes. I apologize for asking you to vote. Oh my gosh. Don't ask me again until next time.
I read a funny joke. It says, "Who's counting all these votes? Is it like the sloths from Zootopia?"
Yes. I saw that. I was like, it's so perfect.
It's just Nevada though. Like, come on, dudes. They got like an abacus over there counting this.
I know, it's wearing me out. I'm just like, come on.
We just need one state. Anyway, okay, sorry, I'll stop now.
No, I'm with you. I'm there. Oh my gosh.
I almost made it yesterday without telling anyone my political opinions. I didn't make it, but I almost did.
Better than me. Every meeting this week I've been like, I am so stressed. And they're like, okay.
And it's like, it's not even just Nevada that's crazy. Like, Georgia's within 0.3%. And then Pennsylvania is somehow getting closer and closer by the hour. And it's like, I'm so stressed out. Me and the boys went from like a gaming group to just, all right, who's watching what live count? Like, who's
Oh no. I know. I am trying my best. Okay, I'm gonna end the poll because I'm gonna end the poll right now. I know, Alan, I know. It's like, oh my goodness. Okay, so I have the poll results. Transcription and translation review came ahead in a very close match. So you can see the results. There you go. Okay. But I think that's good. So what I'm going to plan on doing then is I'm going to go over transcription and translation today. So just like through it. And then we will have time on Tuesday to go over gene regulation. So again, I want to assure you that this doesn't mean I won't go over it. It just means that today we're going to focus on transcription and translation. And then on Tuesday, we'll focus on gene regulation clarification. So, all right. Okay. All right. So that's good. Thank you all for participating. It's the first time I've got one of those polls to work well. So, okay. So before I do that, I do want to just mention a couple of things. So normally on Tuesdays, we kind of go through what to expect for the week. So I do want to just make sure that I do that so that we're all on the same. Oops. So we're all on the same page. Okay. So a couple of things is that make sure you get your assignments in. So one thing that I really, really, really want to mention, and again, like I said, I'm really sorry about emails this week. It's just been, whoa. But what is very important is that if you were not able to submit your rough draft that was due last week. So I'm going to go to the modules here, and I'm actually going to go to week 11, and I'm going to scroll to the bottom. This assignment is where you needed to submit your rough draft. So every person in your group needed to submit this individually. So every person in your group needed to submit this. Again, it should have been worked on as a group, so it should be the same for every single person. But if you did not, for whatever reason, submit your rough draft to this assignment by 11:59 p.m. on Sunday, November 1st, so this past Sunday, you need to email me. If you've emailed me already, email me again, because if you didn't have it submitted by the due date, it's very likely that you were not assigned a paper to peer review, because your peer review assignment was dependent on getting your rough draft submitted to this assignment by the due date. So for conducting your peer review, I just want to scroll up to week 12. So what will happen, I'm going to open this up, is that if you're assigned a peer review, let's see, you'll see like a prompt either from email or on Canvas, and it'll say like something over here. This is where you submitted your rough draft. You'll see a little prompt over here. And what you'll see when you open it up is going to be sort of similar to this. So I apologize for showing an example. I appreciate for serving as an example. But you'll see your assigned peer review pop up like this. So you have two options for how you want to conduct the peer review. You can follow the directions and see it's a document. You can download this document and use the Word review tools, or you can actually use the Canvas review tools to do your peer review in here. That's totally fine as well. Leave comments, whatever. If you do a review in Word, you'll see something a little bit different over here, but it'll say like over here, it'll say like attach file or something. So just like do your review in Word, save it, and then upload it here, just say attach file as a comment. And that way the person who you're peer reviewing will see your comments. If you leave comments using the Canvas review tools, which is totally fine, it's just that when you like download this document, your comments and review won't necessarily show up. And so what I would encourage you to do in that case, just to like show us your peer review. Again, if you do like comments in here, just take like a screenshot or take pictures of your peer review, whatever. So after you do your peer review, whether you do it as Word review and upload it as a comment, or whether you do it here using Canvas review tools, take screenshots, what you need to do then is you're just submitting your peer review to this week's assignment. So you just conducted your peer review in the week 11 assignment. This is week 12, our current week. And what you'll do is you'll go here, peer review of enzymes rough draft. So this is also a really beneficial video if you haven't watched it already. You'll go in here and it'll say up here to like submit. Either if you did your editing in a Word document, submit the Word document here. Or if you did your editing in Canvas, just submit your like screenshots of your peer review. So down here, this rubric is the rubric that we are going to use to grade your peer review. So for example, did your comments include specific suggestions? Did your comments include how to improve, resolving problems with logic, etc.? So this is regarding your feedback to the other person. So I just wanted to clarify that. This is very helpful if you want to know kind of like how to give criteria. This downloadable rubric, which I think is associated with the other one, is the rubric that you can use to grade the other paper. This is the same rubric that we are going to be using to grade your final drafts. So it kind of goes through what we're looking for as far as title, introduction, methods. This is the same criteria we've been using to grade your individual submissions of these as well. So this is the rubric you can use to grade your other groups. This is the rubric we are using to grade your feedback on your assigned peer review. So I hope that makes sense. So again, I'm kind of going to go through this again. I see a question in the chat. I will get to it in just a second. So again, if you submitted your rough draft on time, this is where you will go to access your assigned peer review, or if you got like an email, whatever. But this is where you would click on this assignment and it'll say, you know, you've been assigned a peer review, click on that. It'll take you to the rough draft you've been assigned and you can edit it however you want. And then what you need to do is just to this assignment this week is submit proof, or yeah, basically proof of your review. So you can upload your Word document with all your review comments, or you can submit screenshots of your Canvas editing. Whatever works best for you. If you don't have Word, I would recommend using Canvas tools. They're very easy, but just know that you'll just have to submit screenshots instead of Word document, which is totally fine. If you did not submit your rough draft on time and you're not assigned a peer review, contact me via email immediately. All right, so I'm going to try to get back to you as soon as possible. Again, today is whoa, but I will make sure to get back to you as soon as I possibly can, so that you're able to do your peer review. So, but just be sure to email me. Okay, so I'm going to check the chat. All right. Yep, screenshots as overall comment, that's totally fine. The length of the paper, as for any comments regarding length, so I would defer to your lab instructor. So your lab instructor will be, and so this might vary, unfortunately it shouldn't have. This might vary a little bit lab to lab. And so if you were assigned a paper from like a lab associated with like a different instructor, the length should be the same for all sections. We were trying to get students to stick to like 2 or less pages, but how well those guidelines were being like maintained, I guess is what I'm trying to say.
Yeah, because
yeah,
basically the thing that was like worrying me, because we wrote it to how our instructor said.
That's what I would recommend.
Which was like, you know, the discussion should have been like 3 full paragraphs with, you know, so, and each time she looked at it, she said, yeah, like that's good. You've got everything. That's a good length. So that's why when I emailed you stressed out, I was like, yeah, I just sort of like put everything together into one thing because she said all those individual components were good.
That's fine.
And then when I saw the thing and it was just like 1.5 to 2 pages, now it's like ours is like 5 to 6 pages.
That's okay. So probably what will happen is that, yeah, your peer review may say, hey, you need to cut this down. But if your instructor is like, this is what I want you to stick to, like your instructor is going to be the one grading your final draft.
Okay.
Yeah.
I'll probably email her then and ask for sure.
Yeah, I would. Yeah.
All right. Thank you.
Yeah. And Allie, okay, so same there. Just I have to defer to what your lab instructor told you. They will be the one. Yeah. Shows they're supposed to be submitted twice. There should only be one place. Heidi, email me and I'll try to figure it out. I know I might be a little delayed in my response, but everybody should have only been assigned one peer review. So if you were assigned 2 peer reviews, you only have to do 1 peer review.
No, I know it. I know what she's talking about. I also saw what she's talking about. So I submitted it to both because there were 2 places to submit, but only one gave a peer review. The other one was just 100-point assignment, and I was like worried about there being 100-point assignment with no turn in. Because that's the thing, it didn't show up in the module, but it showed up on the calendar as an assignment due that day.
Okay.
That's where I found it.
Yeah, don't worry about that. We don't have any 100-point assignments right now.
Okay.
Yeah, it's okay. Don't worry about it. I promise. If it's not in the modules, that means like we deleted it, and I'm not 100% sure why it's like still there. Canvas is making me want to love life. Natalie, email me and I will reassign you. Yeah. So yeah, I know it's a little bit challenging. And I think like based on the challenges for this peer review, I've reached out and hopefully we'll have better software for the writing assignments next semester, hopefully. I do love Canvas, but there are some pitfalls to it. This one, peer reviews. But okay, Fernando, email me and I'll try to fix it. Yeah, sorry for the issues. Yeah, we'll work it out. But for that 100-point assignment, I'll look into that. I don't know what's going on. But you did not have any 100-point assignments due at all. So don't worry about it. I know me saying don't worry about it is sort of just like, but really, don't worry about it. Okay, so I hope that clarified a little bit about a peer review. If you did have like issues or anything like that, just email me. My responses will be slow. I sincerely apologize for that, but my goal is to get back to you as soon as possible. I promise to do that as soon as possible. Okay, so, all right. Everything good? Ish? Okay, cool. All right, with that, and with the results of the poll, we're gonna talk about transcription and translation. I'm pumped. Okay, so I do think that we went through transcription decently. So does everybody have a raise your hand if you don't mind, raise your hand or send me like a private box message if you want me to go through transcription again. And just FYI, if I get one person saying they need a transcription, I'm going to go over it, because one person replying that they need transcription means that there are probably several people that need transcription. All right, cool. All right, I've got a couple messages saying we need a little transcription, so let's go through it. Yay, thank you all for commenting and raising your hands and letting me know. Okay, let's go through it. Okay, so I do know that we did go over kind of a little bit about, let me get this all set up. Okay, so we did go over a little bit about, and if you have questions throughout, just unmute yourself or type it in the chat, about what like the entire process of protein synthesis is. And it's going from the genes you have in your DNA to basically all of the stuff that makes you, right? So that's the protein. So your DNA and eukaryotic DNA is divided into little sections called genes. And traditionally, and this is what I do want to clarify a little bit later, traditionally, we think about like one gene encoding for one RNA, and that one RNA coding for one protein. After going through gene regulation, you know that's not the complete story. However, it is a really good basis to build upon. So for the purposes of today, let's think, right, that you have one gene that codes for one mRNA that codes for one protein. So a gene is equivalent to a protein. A gene is like the recipe, written down recipe, and the protein is the cookie, right? The recipe in this case is for a cookie, which is what I would really like right now. So in your genes, you have all the instructions for creating a cookie. You have to make a copy of those instructions. And then you actually have to like bring all the ingredients together and like make the physical cookie. And that's your protein. So what we're doing is we're going from genotype, which is the genes you have, to phenotype, which is the expression of those genes. So the stuff that makes us all up. And that occurs in 2 steps. So this occurs in transcription, which is going from gene to mRNA, and translation, which is going from RNA into protein. So I can't remember, remind me, did I tell you all about my grandma's cookbook? It's okay if I did it at all. Okay, good. I did it. Okay, good. Okay. So, well then I'm going to tell you about my grandma's cookbook. And we're going to go through this step. Okay, good. So, something to note, and let me get back to this. I'm going to go skip ahead because I think it might help a little bit to give a summary of this process. So let me open this up. Just a second. There we go. Okay, I want to show a video of transcription and translation taking place, but before I do that, I want to give you just my favorite analogy for transcription and translation. And so it's my grandma's cookbook. So a little background about me. My great grandma was an immigrant from Germany, and she came directly over here from Germany, didn't speak a word of English, and settled outside of Chicago, where she opened a restaurant called the Forest Inn. So adorably, my dad's name is Forrest. So that's kind of where that came from. But Forest Inn, and she, again, didn't speak a word of English, but she was renowned for cooking. And so that's why she figured I could make some money opening restaurants. It's like way easier back then. So she opened a restaurant. It was a bunch of like really good German food. And when she passed away, I actually inherited all of her cookbooks, which is really great, except for they were all in German. And although I've had multiple opportunities to learn German in my lifetime, I still have not learned German. It's a huge deficit and a downfall about me as a person. Someday I will learn German so that I can read these cookbooks. Although I'm not sure that I will be able to read these cookbooks, because if anybody has any experience with grandma recipes, they are usually ineligible. And this is true. So my cookbooks that I inherited from my grandma, this is not a picture of it, but it looks very similar. The cookbooks I inherited from my grandma are in German and also written like any other grandma, where I cannot read them, even if I could read German. It's like arrows and craziness everywhere. So why does this matter? Well, so she was famous for her cooking. Like, again, she had this restaurant, like a frequent, she also, when she passed, I didn't only inherit all of her cookbooks, I inherited randomly like a ton of fur coats. And I was like, why did she have all these fur coats? And it turns out that she was very close with Al Capone, which is where she got those fur coats. And also randomly a private plane, which we didn't know about. So very close friends. And so when I inherited these, obviously knowing how good of a cook she was, I wanted to make everything, right? I didn't really get to encounter her. She passed before I was born, but she was famous at the restaurant for a lot of things. I'm just showing here cookies, but she didn't really make cookies. One of her things was like beef stroganoff, which was so good. Beef stroganoff. I still love beef stroganoff. But I inherited her recipe for beef stroganoff, but it's written in German in a way that I can't read it, even if I could read German. I still cherish it though. And so in order for me to make her beef stroganoff, what I have to do is I have to take this recipe in German that's written in a wild way and write it into a language that I understand. And for me, my primary language is English. And so I have to write this recipe down in English and also in sequential order. So I understand what to do when. So I have to get it from German and wild to English and ordered for me. And then what I have to do is get it from being words on paper to actual food, to actual beef stroganoff. So I went from words to words, transcription, and from words to actual food. And that's translation. So if you're catching my drift, why I told you all about my past, this is why, because this for me was the analogy that hit home. So this is the same thing that happens in your body with transcription and translation. You go from nucleic acids, DNA, which you can think of as like this recipe in a different language that's, scribbled all over, etc. You're going from DNA to RNA, the same language, nucleic acid, or the same format, words, but a different language, right? From German to English. So still written word, but then getting that written word to actual food, or getting the nucleic acid mRNA to protein, like hair color, eye color, like sound of your voice, everything. That's going from written word to stuff. So that's translation. You're switching, here you're switching language, here you're switching medium. So that's a huge change. So the reason I mention this as well is I get a lot of questions like, why does this have to happen in 2 steps? And the answer is, why do we need to go from DNA to RNA? Why can't we just go DNA to protein? And the answer is, I hope you all know that I love you all so much, right? I'm very protective of you. I think you're amazing people. I'm like, you all are incredible. However, if you came up to me and said, can I borrow your grandma's cookbook? I would say no. But I would say, you can't borrow my grandmother's cookbook that I inherited, but I will make a photocopy of my grandmother's cookbook and give you that. I'm willing to share the recipe, just not willing to share the actual book, right? And that's what I would do. I would give you the mRNA, right? So that's the idea. Like, why do we go from DNA to RNA? Your DNA is precious. In a cell, you only are carrying around 1 copy of the DNA that's used to code for everything that cell does. And that cell is kind of controlling all the cells around it and what they do, right? Tissues rely on collectively, properly functioning cells. And that relies on that DNA being protected. In our cells, it's protected in a nucleus. So we have a lot of mechanisms of protection for it in eukaryotes because that DNA does a lot. But we're protecting it from mutations. We're protecting it from like physical damage. We're protecting it. If the cell needs to make hemoglobin or the cell needs to make something, we're not going to just say like, okay, here's the DNA. We're going to say, okay, I won't give you the DNA. I'm not going to open the DNA. The DNA stays in the nucleus. My grandma's cookbooks stay in the closets, but I will make a copy. And that copy is mRNA. We're not going to make a copy of the entire DNA strand. We're only going to copy what you need, right? If you're like, I want the recipe for beef stroganoff, I'm not going to spend however long photocopying every single page of my grandma's book. I'm going to only copy the recipe you asked for. So that's really what the purpose of transcription is, why we go from DNA to RNA, is so that we can protect the DNA. RNA is same format, like same medium, it's still words, but it's in a different language. This is so that your cell can recognize it, that it's not DNA. right? mRNA is allowed to leave the nucleus. DNA isn't. DNA can't be in the cytoplasm and eukaryotic cells or else it's destroyed, right? If there's DNA in the cytoplasm, it's probably a virus. So your DNA stays in the nucleus. It's protected. So mRNA is in a different language, right? It's RNA instead of DNA because RNA is allowed to leave the nucleus and is allowed to be in the cytoplasm. And then once you get that RNA in, out of the nucleus, then we can use a ribosome. Think of the ribosome as the kitchen, right? The ribosome is the assembly center. This is where we read that mRNA into protein. This is where we go from a recipe to the food, right? It's the kitchen. So that is a hyper long analogy. But to be honest, like that was the analogy that made it click for me, like the why and kind of like what I'm doing. But let's, what I want to do is show you one of my very favorite videos of this process taking place. So I think I linked this video on Canvas, but if I didn't, I will send it to you. But what this video is going to show is the process of transcription and translation. I like this video too because it also incorporates a little bit of the gene regulation part, because you're going to see, if you remember from the gene regulation lecture about transcription factors, it's not just RNA polymerase that binds to the promoter. It's RNA polymerase plus a bundle of transcription factors. And in order for that RNA polymerase to be activated, you need an activator. But the activator binds to the enhancer region, and then the DNA like bends over, and that's what activates RNA polymerase. This video shows that. And so you'll see that process take place, and then you'll watch transcription and translation take place. So I really love this. Another thing I want to also answer before we watch this video is a question I get a lot as well, which makes sense. It's a good question, is whenever we're building nucleic acids, so with transcription, we use DNA as a transcript to make RNA, but we have to like make RNA by like building nucleotides together. So where do those nucleotides come from? And the other question is the same. During translation, we're building a strand of amino acids. tRNAs, like have to bring in those amino acids. Where are these tRNAs coming from? Are they just like floating around? And the answer is yes. You'll see it in this video. Yes, they are kind of just like floating around the areas. You have tRNAs kind of hanging around ribosomes. You have nucleic acids very prevalent in the nucleus. So the machinery and the substrates necessary for these reactions, they are localized to where these reactions are taking place. So yeah, it seems really weird that they're just like floating around, but they are indeed just like kind of hanging around. Okay, so let's watch this take place. I want you to take note of the time. This video is only around four minutes long. There's about 10 seconds that I'll just fast forward through because it's useless, but let's watch this video. It's super good.
What you are about to see is DNA's most extraordinary secret, the innermost workings of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything.
So just to give you, I'm going to stop this a couple times to explain things. So just to kind of show you what this is. So she mentioned the gene is this over to the left, right? So this is your gene, what we want to transcribe into RNA. You'll notice we had something there originally, right? This, it's a terrible angle. This, which is our RNA polymerase. And then all of a sudden stuff like flew in and assembled. And those are your transcription factors. So you had RNA polymerase at the RNA binding site, or sorry, the transcription and initiation site. And then you had all of your transcription factors binding to both RNA polymerase and to the TTh box. So that's what this is. So remember, upstream of this complex, you have your regulatory region. So you have an enhancer region or a silencer region. In this video, where transcription is going to take place. So what we're not seeing is that activators have bound to the enhancer region. So the next thing you're going to watch, so we have our pre-initiation complex formed. We have RNA polymerase, we have our transcription factors. And now what you're going to watch is the activators come and activate this process. And then that's when we're going to see RNA polymerase release and then move along the gene to transcribe.
Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA.
So that means that this is your RNA polymerase, and that is your mRNA, like she just said.
The building blocks to make the RNA enter through an intake hole.
Those are your nucleic
They are
acids.
matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body.
It's a bit like taking the information off a hard disk on a computer and put it in.
Useless information. Okay, so what you are watching again is transcription. What I want you to notice again, this is RNA polymerase. We're making mRNA. She mentioned that's by using nucleotides to take DNA and turning it into RNA. Again, I said like the nucleotides to build this, those are all those little yellow fragments hanging out around, as you can see. And then we're just using that to build mRNA. So same format, right? So we're using nucleic acids to build nucleic acids. It's just we have DNA as a template and we're creating RNA. Right.
When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein.
So again, you can see the tRNAs, yes, they are just like hanging out. The ribosome is moving along the RNA from the 5' to 3' direction. And this will show the 3 sites in the ribosome. You'll be able to see the E, the P, and the A site. And you can see those tRNA entering in the A site, exiting on the E site. And in a minute, you'll be able to see the growing amino acid chain coming out of the P site. So this is your ribosome, this is mRNA. These are the tRNAs bringing in your amino acids. And you'll see the ribosome start to build your polypeptide or your protein.
Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome.
So again, sorry to keep interrupting it, but you can see this is the E site. This is the P site. I know that because you can see the growing peptide coming out of it, P for peptide. And then this would be the A site where you have amino acids like entering.
The RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin.
Okay. Okay, so hopefully that clarified a little bit. I want to take a little second to just kind of like, we can go through each of those processes individually, but I think hopefully that summarized like a process in total for you. So what I want to mention is that I love that video so much because hopefully you noticed that it said this process takes place in real time. And the protein that we watched created was hemoglobin. So from the lectures, you know that hemoglobin is the protein expressed by red blood cells that carries oxygen throughout the body. And that means that that protein is very, 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 very important in your body. And that video was around, aside from that person talking about whatever, that video was around like 4 minutes and 15 seconds, if I didn't interrupt it constantly, 4 minutes and 15 seconds. That means, again, that was in real time. So that means that's how long it takes for your body to go from a gene into a protein. About 4 minutes and 15 seconds for hemoglobin, right? That was in real time. Your body is constantly needing to make hemoglobin. So the reason I like that video so much is that means that while you watched that video of hemoglobin being made, you did exactly what you were watching on the screen during that time. You made hemoglobin while we were watching that in the same time, at the same time. It blows my mind. That's one of my favorite videos to watch anytime because I'm like, man, if I'm having a bad day and I'm like, I can't do anything right, I just watch that video and I'm like, yes, I can. I can make hemoglobin in 4 minutes and 15 seconds. I am doing great. And so you should think that as well, right? What you just did was successfully in 4 minutes and 15 seconds build hemoglobin. How do I know that you did this? Well, if you're here, presumably you're alive. And if you didn't make hemoglobin in that 4 minutes and 15 seconds, I am sorry to say that you probably would not be here right now. You need hemoglobin to carry oxygen. It's absolutely required. So if anything else goes wrong today, just know that you should be very proud of yourselves for making hemoglobin in 4 minutes and 15 seconds. So good job. I'm proud of you. I'm proud of me. Yeah. Okay. So hopefully that video helped a little bit. Let's take a look. Let's go back to this. Okay. I didn't share my screen. Okay, so that is an overview of the processes of transcription and translation, but let's look at this more individually. All right, so what we have to do, as shown in the video, is we have to go from genes to protein, and this means we have to go through transcription and translation. So starting with transcription, and again, realize, now based on the gene regulation lecture, that this, what we're looking at on this slide, is a little oversimplified compared to what actually happens. So in all actuality, right, the promoter is a little bit more complex than just like a little yellow region. So in all reality, you have the initiation site, you have the TTh box, you have up here, you have the enhancer region or the silencer region, whatever. But for the purposes of just, let's talk about transcription, let's simplify it. So what happens first is we have a gene. The gene runs from promoter to terminator, meaning that the shaded area is the region of a gene that we need to turn into mRNA. So what our machinery that's going to help us do this is RNA polymerase. So RNA polymerase binds to the promoter, binds right here, and that's initiation of transcription. Is just that RNA polymerase binding to the promoter. I'm sorry, there is like an airplane flying by with a giant banner, and I got very distracted. I apologize. Okay, so for transcription, we take a region of DNA, which runs from a promoter to a terminator. And we have to have RNA polymerase bind to the promoter, again, because that's the machine that's going to build our mRNA. So initiation, that's all that happens. RNA polymerase binds. And then we have to enter elongation, which is when what we're doing is elongating our molecule of mRNA. So RNA polymerase binds to the promoter. Once it gets the signal to go, it runs along the 3 to 5 prime strand of DNA and makes a copy of that strand that's complementary, meaning that if you use a 3 to 5 prime strand, you're going to create a 5 to 3 prime strand of RNA. So we read 3 to 5 into 5 to 3, and we're reading this blue DNA strand into this pink mRNA strand. And again, all we're doing is we're just using these like little black lines are nucleotides. So if on our DNA we have a C, we build our RNA with a G. If we have a T, we build our RNA with an A. So we do this all via complementary base pairing. So just going along this gene and building RNA. We do this until we reach the terminal region, at which case everything just like disassociates and we free our mRNA. So after that, a couple of things happen. So we have to process this RNA, and we actually do this doing 2 things, and I won't spend too much time on this just because I think everybody kind of gets this, but we just add a cap and a tail, but we also have to splice this molecule. So splicing, I think one of the common misunderstandings, and I'm going to jump over here to discuss this. One of the common misunderstandings of the gene regulation when we talk about splicing is what's the difference between splicing and alternative splicing? And the answer is there is no difference. Like what? I know that's like really confusing. So the reason I say that is because splicing is strictly a process. So in eukaryotes, every single gene goes through transcription. And then if it's mRNA, the transcription actually generates pre-mRNA. Splicing is simply the process of cutting out the introns from the pre-mRNA. So it's just removing the non-coding regions. So splicing is just a process, right? So splicing is simply the process of removing non-coding regions. Alternative splicing is actually just a more complete story of how splicing really takes place. And again, we traditionally think of transcription and translation as having like one gene transcribe one mRNA, and that one mRNA lead to one protein. So sort of what you're looking at here on the left is sort of like our, you know, simplest understanding. But in all reality, as you learned in that gene regulation lecture, the vast majority of genes result in the production of more than one protein. And the reason this is because when they go through splicing, when they go through the process of splicing, what happens is this one pre-mRNA is spliced into 2 or more mRNA. So this is still just splicing, but it's just a way that splicing is used to generate more than one mRNA and therefore more than one protein. So splicing and alternative splicing are the same thing. It's just splicing is the process, right? It's just removing introns. Whereas alternative splicing is how we can use splicing to take one gene and turn it into multiple proteins. It's just by kind of like removing introns and then making different mRNAs. Okay, so let's hop back over here. So we go through mRNA processing, we add a cap and a tail, we splice it, and that's how we get our pre-mRNA to mRNA. And then that mRNA leaves the nucleus. And then once it leaves the nucleus, it heads to a ribosome. So now we get to translation. And I think that this is actually, let me double check that I'm okay on time. I don't want to hold you over time. I think I still have 5 minutes. Let me use them. So my clock is wrong. Do I have 5 minutes or do I have 10? Can somebody write?
You have 10.
Yes. Thank you, Rita. Okay, cool. I have 10 minutes. That's great. Okay. All right. So that mRNA leaves the nucleus and it goes to a ribosome. So I'm going to skip to right here. So this is skipping ahead, but I just wanted to show you. So this is that mRNA we built, this pink mRNA, and you can see it associated with a ribosome. So ribosomes are going to read this mRNA in the 5' to 3' direction. So see how it binds at the 5' end. And the ribosome is going to move down the mRNA this way, but it's going to read this mRNA like nucleotides in sequences of 3 nucleotides. So let's talk a little bit about how ribosomes read this mRNA. Okay, sorry. Okay, so this is looking at your mRNA molecule. So mRNA molecules are arranged into what are called codons. And codons are basically just like, so remember here, what we're trying to do is we're trying to go from the language of nucleic acids, right? So the language, it's like written word, into the language of amino acids or protein. So we're trying to go from written word to like actual food, right? So that requires a lot of ingredients. In this case, the ingredients are like tRNAs and amino acids. And this is all assembled in the ribosome, which is like the kitchen, okay? So as with anything, right, if we need like read the word flour, we know we need to grab a bag of flour. So codons are essentially those words that code for like physical things, right? So these codons basically code for specific amino acids. So I do want to specify a point of confusion has a lot to do with like anticodons. All I want to tell you about that is just like, don't think about anticodons. I know that's a little weird. Don't think about anticodons. You don't need to know about anticodons. The only point of an anticodon is how we know, if we have, for example, AUG, how do we know what amino acid that codes for? Again, so if this word is flour, how do we know like what bag of white powder is flour, right? So how do we know that? And the tRNA are those translators. Okay, so for example, if we have a tRNA, if we have the codon AUG, we have a specific tRNA that recognizes AUG as coding for the amino acid methionine. But again, this requires, like, the tRNA is our translator tool. The tRNA has to recognize AUG is methionine. So how does it recognize that? Everything with nucleic acids, like recognition and building things, is done via complementary base pairing. And the same is true here. So with our mRNA, if we have AUG, this tRNA is going to recognize AUG by having a complementary nucleotide sequence. So that's the only thing you need to know about anticodons, is that they're complementary sequences on tRNA, and they just help us recognize, like, what is this word, right? AUG in nucleic acid means methionine, an amino acid. So it's just our translator tool. So how do we translate that? We use the codon table. So again, if I have a strand of nucleic acids, mRNA, and I divide that into 3 nucleotide sequences or codons, we use the codon table, which the codon table tells us which codons in our mRNA code for which amino acids. So for example, AUG, if I look at my codon table, AUG right here, this codon tells me that means I'm going to make I'm going to bring in methionine. If I have CGA, CGA, that's arginine, and so on and so forth. And that's what I kind of did here. So I see AUG, methionine, CGA, arginine, UCC, serine, GAC. The only purpose of the anticodon is just to help the tRNA recognize that. You don't need to know the sequence of the nucleotides for the anticodons. Just forget it. Just forget about the anticodon. Yeah, okay. So we have our mRNA divided into codons. We need to get our tRNA in there, and we need to assemble our amino acid. So how does this happen? So this is our mRNA. Again, we have our ribosome here. Our ribosome has 3 sites, EPA. The very first, our start codon, is always, always, always going to be the codon AUG. And the tRNA that carries methionine is always, always, always going to bind AUG. The very first tRNA that enters a ribosome always binds to the P site. You'll see why in a sec. So this is the start of initiation of translation. We have our ribosome bound to our mRNA. We have our start codon in the P site. We have our very first tRNA, which is always going to carry methionine, bind to our start codon. And the next thing that's going to happen is, because we have the A site, this is where our next tRNA is going to enter. So we can predict it, right? So we have CCG. Let's see if we can predict what that next tRNA is going to enter. CCG, CCG. The next tRNA that's going to enter is going to carry proline. So again, we will see here, and there you go. CCG means the tRNA carrying proline is recruited to the A site. So now what you're going to see happen is we need to form a peptide bond between these two amino acids. And in order to do that, this tRNA is basically going to donate this amino acid to this tRNA. And we're going to build a bond between those two, a peptide bond. So you're going to see that. There. So again, methionine was just transferred over here. We built a peptide bond between those two. And then the ribosome is going to move to the right. So that will shift this codon into the E site. That will shift this codon into the P site. And that will mean UAC will be the next codon in the A site, leaving space for our next tRNA to enter. And you can see that here. So we have this tRNA carrying the growing amino acid held in the P site. That tRNA pops out of the E site. And now we have space for our next tRNA to enter. Once it does, we're going to be able to transfer this over here, which you see happening. And then that just continues all the way down the RNA. This continues actually only until we get a stop codon in the A site. But once we reach a stop codon in the A site, stop codons don't code for amino acids. They code for release factor, which basically just disassembles this entire thing, which means we release that peptide. Okay, so I know I kind of tried to speed through that a little bit towards the end, but I will post these, actually I might have posted these slides for you last week, with these slides going through this process. My hope is that that video also helped, but if you have questions remaining, I think I'm now officially out of time. My clock is wrong on my computer and I can't fix it because it requires an administrative password. It's like, oh my gosh. But if you have questions remaining, make sure to include them in this week's discussion board so that I can see them. Because I need to know exactly, and try to take the one attempt at the Unit 4 review to kind of see how we're going to be asking questions about these processes as well, transcription and translation, because I want to help your understanding more. I just need to know exactly where the points of confusion are. So if you could just help me that way, that would be a huge help. But hopefully today helped. And what we'll plan on doing on Tuesday is covering a little bit of the questions about gene regulation. And then as well as any questions sort of about like cell cycle and stuff like that. Probably focusing on transcription and translation and gene regulation on Tuesday, since that's the stuff that's on your upcoming checkpoint. And then we'll meet again probably on Thursday and then talk about sort of anything else you need to talk about. What you can anticipate for next week is a very light week, mitosis and meiosis, really fun lectures, and then like 2 labsters. I mean, checkpoint as well, but that's about it for next week. Light week. All right. If you have any questions, I can hang out, but if you don't have any questions, have a wonderful weekend. I'm sending you all the most positive vibes that ever existed.